everyone, and welcome back to this video related to chapter 6.2 of CBSE Biotechnology course, which is related to plant cell culture and its applications. In the last video, we have discussed major events or history which happened during the development of plant cell culture as a technology, its introduction, and some other things related to establishment and minimum requirements for establishing a plant culture cell laboratory, how we prepare media, what are the ingredients of plant growth media, and some other aspects. I'm sure after watching that video, you are able to answer certain questions. For example, what is plant biotechnology? What are the benefits of biotech crops? History of plant tissue culture? What is the concept of totipotency? What are plant tissue culture media? What are their constituents? And some examples of plant cell tissue culture media. And what are the other methods, popular methods for sterilization of instruments, sterilization of media, sterilizations of explant or plant part itself, and what bare minimum requirements are there in a small plant cell tissue culture laboratory. So moving forward, today we will discuss other aspects of plant cell culture technology and its applications. In this video, I will discuss with you concept of totipotency, types of plant cell cultures, micropropagation techniques and steps, major steps in it, haploid cultures and embryo rescue technology, protoplast culture and its applications, and somaclonal variations. So let's discuss first what do we mean by totipotency. I am sure you guys remember that that concept of totipotency was given in 19th century by Professor Philip White, and there are actually three steps, three major events to basically understand the totipotency: differentiation, dedifferentiation, and redifferentiation. So differentiation means starting from a cell that that cell develops into a tissue or into a cell whose fate is decided. For example, if you see here is this plant, there are many organs. Root is an organ, stem is an organ, leaf is an organ, and flower is an organ, and there are many tissues. So these tissues are developed either from shoot apical meristem or root apical meristem. There are two meristems normally available in all plants. Shoot apical meristem, which basically differentiates into growing leaves and stems, and root apical meristem, which makes underground root part. After some time, if it is a flowering plant, then shoot apical meristem also converts some part of it to a flower meristem, and that meristem converts into flower. So the cells in shoot apical meristem is decided, or its fate is decided, that it will make a part of shoot, mostly either stem or leaf. This is called differentiation. That means the totipotent cells which are there in shoot apical meristem, or in other words stem cells which are there, they develop into a part which is required. So their fate is decided. On the other hand, the concept of dedifferentiation says that if we alter the growth conditions, media conditions, and we provide certain growth hormones in abnormal conditions, in abnormal concentrations, then a cell which has already been differentiated can enter into again stem cell phase or dedifferentiation phase. For example, if we take a part of flower as shown here, or if we take a leaf from plant, take it out, sterilize it, and put it into a specialized growth media which is having certain conditions or certain growth hormones along with other nutrients, then it can again go back to previous condition, enter into dedifferentiation condition, and we can basically decide that how we can use those leaf cells to produce more plants. So the turning back of a differentiated cell into a dedifferentiated state or a stem cell state is called dedifferentiation. Again, if we change the conditions, again we change hormone conditions, we change growth conditions, then these dedifferentiated cells can again be made to develop into a whole plant or a whole organ, and then this process is called redifferentiation. So the whole thing of totipotency is basically relied on three processes: differentiation, dedifferentiation, and redifferentiation. In the next few slides, I will discuss how actually it happens. So the major steps in micropropagation, as I discussed in last video also, depend upon three major uh, considerations related, which first is a plant part that we are going to tissue culture. Second, a suitable growth media need to be decided that this plant part from this plant is can be grown on a defined suitable media. And third, a growth condition. So for a hypothetical experiment, let's say we are going to tissue culture shoot apical meristem for a popular plant or any flowering plant. So what we will do, first we will isolate the shoot apical meristem. As you can show here, shoot apical meristem looks something like this. This brown part is actually the cells or stem cells which differentiate into these leaf-like structures. So these are the leaf primordia or flower primordia if in case we are talking about flower meristem. So these are the ones which are developing. But these cells here, which you can see in this light brown spot, these are the cells which are actively dividing, and then they give rise to the newly developing tissues. So we basically isolate it and then we sterilize it. I'm sure by now you remember how we sterilize explants. Mostly we sterilize explants through a mild washing with this day-to-day bleach we use in home, sodium hypochlorite, 1 to 4% normally we use for sterilization of our plant parts. Then we put under sterile conditions in laminar flow hood, which it's a bench which provide us sterile air. We put shoot apical meristem on a suitable media, and the media is sterilized either by autoclaving or by filter sterilization, and it is solidified by a suitable gelling agent such as agar or phytogel. After some time, we provide growth conditions which we have already standardized, either the light conditions and dark conditions and temperature. The shoot apical meristem starts proliferating into many plantlet-like structure, and those plantlets then put into rooting media. Rooting happens, and afterwards these individual rooted plantlets are transferred to soil initially in pots, and later on they are transferred into fields. So these are the major steps in micropropagation. Now, how many types of cultures are there? As far as our tissue culture is concerned, normally we talk about two types of culture, organ culture or explant culture. If you see this plant, there are many organs, as I discussed in previous slide also. Root is an organ, leaf is an organ, stem, internodes, these are all organs, lateral buds, these are organs, and flower is an organ. So depending upon what type of plant we are interested for micropropagation or tissue culture, we take a suitable growing or healthy tissue from a plant part, and then we do its culture, and then it's called simply an organ culture. On the other hand, if we whatever organ we take or whatever tissue we take in terms of plant tissue culture, after starting plant tissue culture, we call it as an explant. So if we are taking leaf organ as a tissue culture, then it's leaf organ culture and leaf becomes an explant. If we are taking flower for plant tissue culture as an organ, then it's flower organ culture, and flower is an explant in the tissue culture pipeline or that tissue culture experiment, and so on. So we can take basically we can do leaf culture, flower culture, stem culture, or parts of the stem, internode culture or nodal culture, or there are these axillary buds, we can do axillary bud culture, and we can also do the seed culture, because there is embryo in seeds, and embryo is again a living part which can be used for plant tissue culture purposes. As far as plant tissue culture as a process is concerned, there are two types of cultures: direct plant tissue culture, which is also called organogenesis, and indirect plant tissue culture, which is also called somatic embryogenesis. So here is it's a hypothetical example for organogenesis or direct tissue culture. What we do here, we take any suitable organ. So here, as illustrated, either you take leaf disc or an axillary bud or an internode from the stem, for plant tissue culture. So we take this, we sterilize any of these tissue, and then we put them in plant tissue culture media for for specified growth temperatures and other conditions, and then it develops directly into plantlets. As you can see here, the node, there are many plantlets coming out from the internode, and then there are in this example many plants are coming out from leaf disc. So this thing is called organogenesis. That means we are taking organ as a culture, as an explant, any of the suitable organ, and it is developing directly into plantlets. It is organogenesis. On the other hand, the somatic embryogenesis or indirect, we will discuss in next slides. So again, to illustrate about organogenesis, this is an example which we are doing here in IIT Roorkee, which relates to the tissue culture of lemon and making transgenics in lemon for certain functional genomics purpose. So these are the lemon seeds, as you can see here. We basically germinate these seeds in normal light and dark conditions. After the seeds are germinated, and for example they are 10 to 15 days old, you can see there are these tiny stems. We cut these stems into different small pieces, and these are called epicotyle. So the first stem which comes out after seed germination is called epicotyle. So the epicotyle is cut into small pieces from say for example from one seed, an epicotyle which is 4 to 5 centimeter in length, we cut it into 3 to 4 or 5 small pieces of say for example 1 centimeter length. We put it after sterilization, we put it onto a proper growth medium which is having a high concentration of cytokinin, and after some time we can see that small tiny beautiful plantlet starts coming, and after some more time each explant is giving many each apical meristem, as you can see here, is giving many plantlets. Afterwards, these are put onto a different media. Here, we basically decrease the amount of cytokinin and we increase the amount of auxin. And in these conditions, these regenerated plantlets, they start making roots. And when they have enough roots, we put them into small pots with soil, and when they grow for they have many leaves, they grow in height. I mean, these are the visual observations, they are healthy, and then we put it into soil, and after some years they start making fruits. So this is a general organogenesis method for citrus, and this method can be applied for many other similar plants, say for example oranges or any other fruit plant. The same method can be applied for getting disease-free plants. On the other hand, somatic embryogenesis is indirect culture. What we do here, we take our explant, just like we take a leaf explant or apicotyl explant in the last slide. So when we take explant here, rather than going for direct organogenesis, we change the condition in tissue culture media and we provide a much higher amount of auxins, so that the explant cells, they start dividing very rapidly, and then they make a kind of unorganized mass of cells which is called callus. So these are basically the explant cells are undergoing de-differentiation, as we discussed in previous slides, and they make an unorganized mass of cells which is in which cells are dividing very fast, because we have provided lots of amount of auxins, and auxin induce cell division, uncontrollable cell division in other words, and it makes an unorganized mass of live cells which are dividing very fast, and this is called callus. So you can see here a leaf disc is isolated. It is basically grown on a suitable media. Most of the time it is the media is very having high concentrations of auxin. It makes a kind of unorganized mass of cells. This is called callus, and afterwards we again change the condition of nutrient media. Now we remove auxins and we provide a higher amount of cytokinin, and from these cells new plantlets comes. So and in fact the number of plantlets are generally more than organogenesis. The callus cells basically make on the surface, they make somatic embryos. What is somatic embryo? As the term indicates, it is a non-zygotic embryo arising from the somatic cells. So the somatics, these are called somatic embryos. However, they, in other words, they are just like the zygotic embryos, provided suitable conditions. Like the zygotic embryo, they also germinates and they make roots as well as shoots, and these plantlets are then transferred into potted soil and then into fields. One special thing about indirect culture, that it is very useful in making transgenic plants because of the callus phase which is there. This is an example, a personal example. Here we did basically plant tissue culture in three different species of wheat, that is triticum. So the three species are bread wheat, Triticum aestivum, pasta or biscuit wheat, Triticum durum, and other wheat is Triticum dicoccum. So in all these three cases, different explant was taken for indirect or somatic embryogenesis. In case of Triticum dicoccum, immature inflorescence was taken, immature flowers were taken for as an explant source. In case of Triticum aestivum, immature embryo was taken as an explant source, and in case of Triticum durum, it's a mature embryo from dry seeds was taken as an explant source. So if you see on the surface, these round structures, these are somatic embryos. These round structures, beautiful round structures, these are somatic embryos, and here also. And when we change our condition, we provide cytokinin in the media, then these round structures, basically they start developing into different plantlets, and you can see there are many plantlets coming out of one single callus, and each plantlet after some time has its own root, and then they are transferred into potted soil and into the fields. So this process is called plant regeneration through indirect or somatic embryogenesis way. Afterwards, let's discuss some applications of popular applications of plant cell tissue cultures after going through the methods which are available to us. So first and foremost method of plant cell tissue culture or micropropagation is basically development of virus-free plants. So you need to listen carefully. I am saying virus-free plant. We are not saying virus-resistant plant. What happens? It has been seen that if a plant is infected by a virus, then all plant parts are after some time all plant parts are heavily infected by virus except the shoot apical meristem or the root apical meristem. And shoot apical meristem, since it is available, it's an above ground tissue, it's easy to basically locate in different branches shoot apical meristem. So what people do, they just isolate shoot apical meristem because it is devoid of any viral infection. Again, do the tissue culture or organogenesis or indirect, whatever the method of choice, depending upon what is our purpose. So in here, I'm showing direct organogenesis method, shoot apical meristem. It is cultured on a suitable media. It makes many plantlets. Those plantlets are basically first rooted and then transferred into soil and then to a farmer's field. So these plants, since they are developed from a shoot apical meristem which cannot be infected by virus, these plants are virus-free plants. So for example, if we have an elite variety or an endangered germplasm which is heavily infected by virus, then and we need to basically save it. So take its shoot apical meristem, do its culture, and then multiply the regenerants, and that way we can have many plantlets from that virus infected plant, and these plantlets are virus free because they are coming from virus free shoot apical meristem. This method is mostly very popular among the plant species or crop species where we do not see seed formation very frequently. Say for example, potato, banana and sugarcane. Most of the time the seed material or the growing material which is distributed to farmers field, they are generated by plant tissue culture methods in potato, banana and sugarcane, and many a times it is basically developed from shoot apical meristems only. Another application is production of artificial or synthetic seeds in plant tissue culture. These are the beautiful picture. I have sourced this picture from one of the book. The source is given here for the book. So what we do, or why they are called artificial seeds, because in these we basically take a plant explant, a suitable explant depending upon different plant species. So, for example, take shoot apical meristem for banana and take shoot apical meristem from banana from many hundred plants. It will not matter whether the plants are diseased or not, they are virus infected or not, because shoot apical meristem will remain non-infected by virus, and cover those shoot apical meristems or those plant parts in a layer of nutrient media covered by a solidifying agent, which is in this case either calcium alginate or sodium alginate. You can see here these are the small tiny balls, and each ball is having one explant in it, and you can transfer these balls to their destinations laboratories, and then they can take it out from the calcium alginate ball and put again on a suitable media where it will germinate and make a new plant. So for example, if let's discuss about potato. So Potato Research Institute in India is in Kufri, which is near Shimla. And say for example, if Indian Potato Research Institute made a new variety which is resistant to bacterial diseases or viral diseases or fungal disease, or for example it is high in nutrients or it's very high yielding, and it needs to be sent to different parts of our country, for example southern parts, western parts, and other parts very fast. So what people can do in that laboratory research institute, they will basically take many plants of this improved variety, collect shoot apical meristems from them, make alginate balls, and then they can be shipped via normal post to different parts or different agriculture universities of India, and then there they can be grown again and can be distributed to farmers. For example, same thing can be done in reverse order. Say for example, Indian Sugarcane Research Institute is in Coimbatore, and they made some good varieties in Coimbatore, and then these varieties or these germ plants need to be transferred to northern part of India. Same thing can be done in the same way. So the process by which we take an explant, convert it into a nutrient media, fill balls of calcium alginate, then it is called artificial seed. So basically it is called artificial seed because it is having a starting material for a plant. Now these artificial seeds can also be used for germplasm conservation, which is again one of the application of plant tissue culture. If we need to basically conserve endangered species or elite germplasm or important plant species for a very long time, then we can do it in two popular ways: through plant tissue culture, make artificial seeds and put those artificial seeds in ultra low temperature. Most of the time it is provided by liquid nitrogen, which has a temperature of minus 196 degrees centigrade, along with some cryopreservants such as dimethyl sulfoxide, properly called as DMS, so or different concentrations of glycerol, and we can basically indefinitely keep these artificial seeds for indefinite time in those conditions, and as and when required they can again taken out from cryopreservation and can be used for normal plant tissue culture based micropropagation. The second method is a short-term storage of germplasm, which is cold storage. So in this case, if we need to store our germplasm only for a few months or a few years, then we can put them these artificial seeds at a temperature which is around 4 degrees centigrade, and as and when required, take it out, and we can do normal plant tissue culture practices with these artificial seeds. And this way we basically use plant tissue culture methods for germplasm conservation. Here I would like to basically attract your attention for a general knowledge thing. Globally, there is a program going on to save maximum as much as germplasm from our planet to be conserved, and a famous facility is coming which is also called the Svalbard Global Seed Vault. So I suggest you as a general knowledge enthusiast that you study what is Svalbard Global Seed Vault and what it is doing as far as germ plant conservation is concerned, and India is also part of it. Another application of plant tissue culture is in the development of haploid plants or haploid culture. So, as the term suggests, haploid means the having one set of chromosomes only. So, I'm sure by now you know what is the normal. Each cell has two sets of chromosome, and then the cells are called diploid cells. Haploids are the one which has only one part of the one set of each chromosome. So normally these haploid cells or the whatever is developed from haploid cells in plants, there can be two sources of haploid cells. One is the male haploid cells which are pollens, and other is the female haploid cells which are basically ovules. So if we do tissue culture either by using pollens or by ovules, then if they regenerate, then the resultant plants which regenerates from pollen culture or ovule culture are haploid. They can normally do not make their offsprings by themselves, but they are important for certain purposes. Say for example, uh, I'm sure you know in by studying genetics that the traits are either dominant or recessive. That means either they expressed and they are visible, and either they are expressed but not visible, which is called dominant and recessive respectively. In case of haploid, whatever trait is there, whatever allele is there, either it is a dominant, it will express, but even if it is a recessive allele which is coming out from a plant developed through uh, pollen culture or ovule culture, it will express because it has only one copy of that allele, and even if it is recessive, it will express, and this way we can see the effect or phenotype of the expression of a recessive allele. In this way, haploid plants are very important. So there are some terms as far as haploid plant development are concerned. Anther culture, which is called androgenesis. Ovule culture, which is called gynogenesis. That means if we are taking anthers, what are anthers? Anthers are the tissues where pollens are there in a flowering plant. So there are two ways. Either we tissue culture anther itself, or we tissue culture isolated pollens from the anthers. So if we are doing anther or pollen culture, it is called androgenesis, and if we are doing ovule culture, it is called gynogenesis. Most of the time, for haploid production of haploid plants, anther culture or pollen culture is preferred rather than ovule culture, because the numbers of pollens are thousands and millions in most of the plant species. On the other hand, ovules are in limited numbers. And all the major crop plants like rice, wheat, barley, etc., they have been the good protocols are available by which we basically do anther culture or pollen culture for uh, haploid plant production. And I'm sure you know that there in flowering plants or in angiosperms, when sexual reproduction happens, so there is a term called double fertilization which happens. So fertilization happens actually two times. First fertilization basically give rise to the embryo, and second fertilization give rise to the endosperm in the seed. So endosperm is a triploid tissue, and then endosperm is basically meant for uh, uh, the food source for the growing uh, embryo. So in case sometimes we also culture endosperms in plant tissue culture, and in this case the resultant plants are triploid plants, because endosperm is a triploid tissue uh, because it results from double fertilization. These are the examples, as you can see in this picture: androgenesis, where either anther culture is done in case of wheat or pollen culture is done in this case. So in both the methods you get somatic embryos, and then these somatic embryo germinates into whole plants. For haploid production or other molecular breeding purpose of plants, these days we also basically do interspecific crossing or interspecific embryo formation for different crops. Say, for example, if you see this example, these are the here what scientists are trying to basically study the effect of wild wheat genes if they are introgressed or if they are introduced into cultivated wheat. So what they do, they take two different species and they try to basically uh, do the crossing between male and female of two different species. In this case, what happens, double fertilization does not happen due to some barriers. Only a pseudo or a haploid embryo is made from one of the plant species, and to save this embryo, because there is no endosperm, as I said, double fertilization does not happen because of some barriers. So this embryo cannot grow indefinitely because there is no food available, there is no endosperm available. In this case, what scientists do, they take out these immature seeds. You can see that there are certain embryos here, tiny embryos, and the seed is hollow. This is a normal seed, and this is interspecific crossing seed. So interspecific crossing, there is no endosperm. So what people do, they take out these seeds into laboratory, sterilize them, and under sterile conditions in a laminar flow hood, take out this tiny embryo, and this tiny embryo put on a growth media where it can grow. So this process either also used for haploid production, and this process is called embryo rescue. Isolation of an embryo where no endosperm is there, and if this embryo needs to be saved, take it out under sterile condition in a laminar flow hood, put it on a media, and then it develops into whole plant. This is an example of embryo rescue. Another thing which is very useful for gene identification and their function is protoplast culture. So uh, protoplasts are the plant cells without cell wall. So I'm sure you know that plant cells are special than other organism cells, microbes, and animal cells or other cells. That plant cell has an extra covering which is mostly made up of cellulose. This is called cell wall, and that's why plant cells are more rigid and they have more shapes in comparison to animal cells or microbial cells. So whatever is inside the cell wall is called the protoplast. So what scientists does, and these protoplasts are very important for ascertaining the functions of different genes, and they can be basically merged with different cells. I mean, there are many applications for these isolated protoplast. Isolated protoplast looks like a three-dimensional ball, and different organs are visible here. As you can see, this is a single protoplast, and these are chloroplasts, which are visible here, the green color dots. So to isolate protoplast, what normally it is done that normally green tissues and leaves are taken, they are chopped into small pieces, and then they are given a liquid media which contains cellulases, hemicellulases, and pectinases. So these are the major constituents of plant cells: cellulose, hemicellulose, and pectin. And these enzymes degrades cellulose, cellulases degrade cellulose, hemicellulase degrades hemicellulose, and pectinase degrade pectin. And when these are degraded, that means cell wall collapse and the entire protoplast comes out in the media. This protoplast can be used for many purposes such as biochemical and metabolic studies related to gene functions, cell suspension culture. Again, cell suspension culture is also a type of culture where rather than culturing plant organs or plant parts, we take protoplast and culture protoplast, and these protoplast can develop into resultant plantlets, thousands and thousands of plantlets if the protoplast culture is successful. But the major use of protoplast culture is the fusion of different species to make somatic hybrids. So uh, as I discussed with you in the previous slide, embryo rescue, another method of making interspecific hybrids is somatic hybrids through plant protoplast cultures. Say, for example, take protoplast from one species of a plant and take protoplast from another species of plant, mix it somewhere in a suitable growth condition and liquid media. there are chances that it will mix up and make one single protoplast, and then it can be developed into the whole plant, and those plants will called as somatic hybrids, because the the protoplast is coming from two different type of species. Same way, if protoplast fusion is done in one nucleated, that means complete protoplast, and one enucleated protoplast from a different species where nucleus is taken out, these resultant plants will called cytoplasmic hybrids or hybrids. We can also do genetic transformation by using protoplasts. Take protoplast of a species and inject it with certain genes from other plant. These genes can get incorporated into the genome of the plant where the protoplast is taken, and then this protoplast is transgenic protoplast now. And if plants are regenerating from this protoplast, then it's a method of regenerating transgenic plants. So as I said, somatic hybrids gives us many possibilities. These are the two examples which I basically want to discuss with you. In this case, a cultivated brinjal variety species is taken, and the variety is basically uh, sensitive for certain diseases, and the same time there is a wild variety available, wild species is available which is resistance. You can see it here. The cultivated one has a larger fruit size, the wild variety has smaller but more number of fruits, and the somatic hybrids, when it is made by isolating protoplast from cultivated species as well as from the wild species, and somatic hybridization was done by protoplast fusion, the resultant variety has larger fruit size as well as disease resistance. Same way, I'm sure you heard about this pomato. So pomato is a somatic hybrid which is again made by two different species of Solanum. Solanum tuberosum, which is potato, and Solanum lycopersicum, which is basically tomato. So what happens here, two different plants, protoplast was isolated and somatic hybrids were made. So basically you can have two different type of fruits in one single plant. In the underground part, the plant makes potatoes, and on the above ground part, the plant makes tomatoes. So and other names are also given for such type of plant, like brassico raffalo brassica, which is basically again has two underground part has radish, and above ground part has cauliflower. And then there is tomatina, again the same way. Tomatina has two different types of fruits, underground and above ground. So these are examples of somatic hybrids. Tissue culture is also used for the production of secondary metabolites. I'm sure you know what do we mean by primary metabolites and secondary metabolites. So primary metabolites are those chemicals or metabolites inside plant cells which are basically required for normal growth and development. Secondary metabolites are those which are not absolutely required for growth and development, but under certain conditions they provide benefits to the plants. Many a times these secondary metabolites are also useful for humans. Say for example, this azadirachtin. So azadirachtin is an alkaloid which is which is found in the leaves of Azadirachta indica. That means the neem. Neem tree, the botanical name for neem is Azadirachta indica, and we all know that neem leaves are basically good as insecticide. The oil from neem leaves it has good insecticidal properties and other uh, insect killing properties. Capsaicin is taken again a secondary metabolite from capsicum. Codeine is a secondary metabolite which is taken from papaver species. That means papaver is this poppy. And there are others listed here. Quinine is a famous secondary metabolite which was used or still is used for it's an anti-malarial drug, and it is again uh, produced by a plant called Cinchona officinalis. So, the how plant tissue culture is helping. So, if we many times what happens, the plant producing the beneficial secondary metabolites, either they are very small, or even if the plant is big, the tissue which is making that particular secondary metabolite is very small, and we need lots and lots of plants for a meaningful isolation of those secondary metabolites. And in this case, those plants need to be grown on a large area of land. On the other hand, if we can make a suitable plant tissue culture method for that particular plant species, say for example, if quinone is isolated from the quinone is isolated from the bark, by the way. So if we can have a method, so say for example, if we find a suitable cell from cinchona plant and do tissue culture and make callus out of it, so there is a chance, or there is a possibility that the callus itself is making quinine inside it, and this can be applied to other secondary metabolites also. Say for example, capsaicin, codeine, dioxygenin, and other like Taxol and vincristine. Vincristine is an anti-cancer drug. It is found in Catharanthus roseus, which is in Hindi is a popular plant date which we found in our homes, Sadabahar. So, but again, to produce vincristine, we need lots of hundreds and hundreds of Catharanthus roseus plants. But if we can establish in plant tissue culture method, or we make callus or organs inside plant tissue culture vials even in a limited space, we can have an indefinite supply of vincristine through plant tissue culture. So in this way, secondary metabolites and medicinal secondary metabolites related to pharmacological, medicinal, or industrial purpose are produced by plant tissue culture. These days, efforts are also going on to produce certain vaccines in the plant cells, so that if we eat those plant fruits, then we basically ingest vaccine at the same time. One of the most important uses of plant tissue culture is the induction of somaclonal variation. So, I'm sure you remember that in the first video I said, when we produce plants through plant tissue culture, they are true to its type. That means they are basically always same as the mother plant from where the plant tissue for tissue culture or explant is taken. However, if basically we do plant tissue culture for a very long time, or if we change the growth condition and we provide abnormal conditions, or certain conditions of growth hormones, concentration of growth hormones, then the resultant regenerants many times is not 100% like the mother plant, and in this case variations are happening, and those variations are called somaclonal variation. This term was coined by Professor Larkin and Scowcroft, and somaclonal variation is defined as genetic and phenotypic variation among clonally propagated plants from a single donor clone. And since this variation is happening from the vegetative part or soma part, then it is called somaclonal variation. Initially, it was in plant tissue culture, it was found that if tissue culture process was done for a very long time and the regenerants are not taken, then variation happens. But after some time, it was found that this can be basically a useful, very useful method to induce variations or genetic variations in some plant species where we do not have regular sexual fertilization or seed formation. Say for example, potatoes. Say for example, banana. I'm sure you have never seen seeds of banana and potato. So in molecular breeding of banana, somaclonal variation, or molecular breeding of potato, somaclonal variation is a very important tool for inducing or for generating new varieties. So how it is done? It's technically it's a very simple process. You take a suitable explant from a target plant species. So for example, banana take shoot apical meristem and make a callus for shoot apical meristem, and then grow this callus for a very longer time, and you provide the target selection. So if we are interested in a banana plant which is salt resistant, which can grow on degraded soils or degraded fields where in the soil salt concentration is more, then try to grow the calli, hundreds and hundreds of calli, on a tissue culture media which has abnormally high conditions of of salts. And if some of the calli which are regenerant, which can grow, which doesn't die, that means these calli already undergone some genetic changes, changes or mutations inside their callus cells, and they are not dying. Regenerate the cells from that calli through somatic embryogenesis, and there is a very high chance that these regenerants which are coming from salt tolerant calli, they plant also are salt tolerant, and then test these salt tolerant plants in actual field conditions, and if they are resistant to high salt concentrations or degraded, they can grow in degraded soil. That means we actually made a new variety of our target plant through some clonal variation, and it can be released for farmers or other purposes growing. This way, there are many applications for somaclonal variation, such as somaclonal variation in crop improvement, development of new variants retaining all the favorable characters along with an additional useful trait. As I just discussed a hypothetical example, that the banana varieties which has high yield, which has good fruit shelf life, but which is otherwise very salt sensitive, can be made salt tolerant through induction of some variation in tissue cultures. Development of elite germplasm and commercial cultivars, the same way which I just discussed. In vitro selection of herbicide, disease, insect and salt resistant somaclones, and breeding of ornamental and tree species. So, somaclonal variation again basically finds its application where sexual fertilization is not very easy to achieve, where there is an important plant species which doesn't make seeds. Then the tool which by which we can do molecular breeding is somaclonal variation. So one example can be large trees which do not make flowers or seeds for example 20-30 years of their life, but they needs to be protected, and they needs to be new varieties are needed in those tree species. So just take shoot apical meristems and induce somaclonal variation by providing specific tissue culture durations and growth hormone conditions, and we can get variations in regenerant plants that would be basically due to somaclonal variation. Here in this picture you can see some successful examples of somaclonal variation I listed in this table, and these pictures also you can see these are orchid flowers, and through somaclonal variation different forms of orchid flower are generated from normal plant. Here the size of petals is bigger, and in these two examples you can see that like here not only the size is increased but the petals become wavy. Here the size is decreased but the petals become wavy, which basically looks good. And this again a salt tolerant variety in wheat, and different varieties of potato developed through somaclonal variation. Some of them are listed here. There are certain disadvantages of somaclonal variation. One biggest disadvantage of somaclonal variation that it is unpredictable and uncontrollable. Basically, we rely upon a chance event that during prolonged tissue culture or callus phase, some cells would go under mutations or other genetic changes, so that somaclonal variation is induced. Somaclonal variation is also highly genotype dependent. Not every genotype of a given plant is responsive for tissue culture purposes. And many a times it has been found that somaclonal variation is not always heritable or stable, so that whatever generation we have, it stays there, and then it cannot make further seeds in which it can be inherited. So basically we need to keep a pipeline always for inducing somaclonal variation that way. It can also be pleiotropic. Pleiotropic means more effects than we desire, and we are looking for, say for example, I discussed a hypothetical experiment where we are looking for salt tolerance. So we got salt tolerance, but the size of fruit is reduced. Again, it is not very well received. So this way it can also be very pleiotropic, and it is disadvantageous. And other two disadvantages are requirement of manpower as well as other inputs, because we need to tissue culture of our explant for a very longer time, and we need to provide growth media time to time. And then technological limitations such as maintenance of uniformity in culture. That means sometimes, since we need to keep the cultures for a very long time, there are practical problems may arise after some time. Uh, the explant can die, or more than desired changes happens in an explant. So with this, this is the end of this video. And again, I framed some certain questions which you should be able to answer after watching this video. Some of them are: what is a concept of totipotency? What do we mean by differentiation, dedifferentiation, and redifferentiation? What are the major steps in PTC, and what types of PTCs are there? PTC means plant tissue culture. What is the difference between organogenesis and somatic embryogenesis? What do we mean by shoot apical meristem culture, and how we basically develop virus-free plants through shoot apical meristem culture? How we produce haploid plants, and what is the use of haploid plants, and what do we mean by embryo rescue? What is protoplast culture and formation of interspecific hybrids such as somatic hybrids or cytological hybrids or hybrids? How we can use plant tissue culture in germplasm conservation? What do we mean by artificial seeds? And what is somaclonal variation? How we achieve somaclonal variation? How it is useful? And what are its disadvantages? With this, I'm sure you are able to understand chapter 6.2 very easily after going through these videos. And in the next video, I will discuss with you methods available to us for transferring genes into plant species, or in other words, how basically we make transgenic plants for different purposes, and then what types of transgenic plants are being produced in in the world as well as in our country, what are their uses and how they are beneficial to us. Goodbye, take care, and go through the video again.